Welcome to the Evolution of Immunity module. In this talk, we're going to learn about the defense mechanisms employed by invertebrate animals. We will begin learning about the immune system in higher organisms, followed by different mechanisms that prokaryotes and protists use to defend themselves. We will then learn about the mechanisms evolved by invertebrate animals to defend themselves. The immune system in higher organisms provides protection against a wide variety of pathogens that can include viruses, bacteria, fungi, protists, and helminths. They're able to do so by employing a wide variety of strategies, which includes the production of many types of antimicrobial compounds, the synthesis of different proteins that have the ability to recognize pathogens, and the evolution of mechanisms to make variations of the same type of protein that enables the recognition and destruction or elimination of different types of pathogens. The immune system of higher organisms also has an elaborate mechanism to retain the memory of pathogens. Such a highly complex system is a product of millions of years of evolution. In order to understand how such a complex system has evolved in higher organisms, let us first look at the different defenses that lower organisms or simpler organisms like prokaryotes and protists have evolved. Prokaryotes are the earliest living forms that evolved on Earth, and they can be grouped in the kingdom of eubacteria and archaebacteria. Prokaryotes are unicellular, and they do not have membrane-bound organelles inside their cells. They compete with each other for space and nutrients, and so they have evolved different mechanisms to protect themselves from other organisms. These include the production of toxins to prevent the growth of other types of prokaryotes or even simpler eukaryotes. Additionally, prokaryotes can be infected by viruses, and thus they have mechanisms to protect themselves against viruses. This is usually done by preventing the absorption and entry of viruses, synthesizing enzymes like restriction endonuclease that can degrade viral DNA, disrupting the viral life cycle, and evolving the CRISPR-Cas system, which is a system that is able to not only protect against viruses, but maintain memory of viruses that have infected the organism previously. After prokaryotes, protists evolved, which are eukaryotes that evolved from archaea. These organisms belong to the kingdom of Protista, which is the most diverse kingdom of life. Protists have evolved different mechanisms of protection, which include producing toxic substances that can prevent the growth of other organisms like prokaryotes. This is an evolutionarily conserved mechanism which we see in prokaryotes as well. Protists are eukaryotes, and hence they have membrane-bound organelles like lysosomes. Lysosomes have hydrolytic enzymes that can be used to degrade and eliminate any type of microorganism that is inside the protist. Protists also produce extrasomes, which are membrane-bound vesicles that have compounds in them that can be delivered to the environment by exocytosis and defend the protist in that manner. Protists also have the mechanism of RNA interference, which is similar to the CRISPR-Cas system found in prokaryotes that enable to protect the protist against viruses. Some protists evolved to form organisms that are now grouped in the kingdom of Animalia. All animals that are in the kingdom of Animalia are multicellular organisms, and they are made up of cells that lack a cell wall. All animals are also heterotrophs. Animals can be broadly classified into two main groups. the invertebrates that evolved first and the vertebrates. In case of the invertebrates, these are organisms that do not have a vertebral column, and there are many different types of invertebrates. The earliest invertebrates that evolved were the sponges. Vertebrates, on the other hand, are organisms that have a vertebral column, and there are different classes of vertebrates. Fish are the vertebrates that first evolved from the invertebrates, followed by the amphibians that evolved from fish. Reptiles evolved from amphibians, and then some reptiles gave rise to birds, while other reptiles gave rise to mammals. Invertebrates were the first animals that evolved from protists. They are multicellular and are much more complex than protists. This allows them to be targeted by other types of organisms, and thus they have to evolve different protective strategies to defend themselves. Some of these strategies are evolutionarily conserved, like synthesizing toxic substances. These can include antimicrobial peptides like defensins. Defensins are able to interact with the plasma membrane of pathogens, and they result in the formation of pores. Thus, the integrity of the plasma membrane is compromised, and the pathogen is now more susceptible to die. Another substance that interferes with pathogens is an enzyme called lysozyme. Lysozyme is known to act on the peptidoglycan of bacteria. Peptidoglycan is a rigid structure that is present as part of the cell wall of bacteria. Lysozyme is able to act on this rigid peptidoglycan, and it cleaves it, thereby causing the cell wall to be more unstable, and thus the bacteria is now more susceptible to die. Other strategies that invertebrates employ are RNA interference, which is also an evolutionarily conserved mechanism. Invertebrate animals have also evolved pattern recognition receptors, which are proteins that can be either secreted into the environment or remain on the membrane of cells or be intracellular and thereby remain in the cytoplasm. The pattern recognition receptors are able to recognize components of pathogens called pathogen-associated molecular patterns. Pathogens can have a variety of pathogen-associated molecular patterns that include polysaccharides that can be found in fungi and bacteria, as well as lipopolysaccharide, teichoic acids, and nucleic acids. Each pattern recognition receptor is able to sense a different type of PAM, and this way the host cells are able to detect the presence of pathogens. In addition to pathogen-associated molecular patterns, there are pattern recognition receptors that are also able to sense damage-associated molecular patterns, which are basically injured host cells or host tissues. Another strategy that invertebrates have employed is the evolution of cells that are able to move throughout the body and protect different parts of the organism. An example of such cells in insects are the hemocytes. It should be noted that plants did not evolve such a strategy, and hence we normally don't find cells in plants that are able to move to different parts of plants. And thus this seems to be more unique to the animal kingdom. Invertebrates have also evolved genes that are able to give rise to many different isoforms, and this helps in the elimination of the pathogen. A well-studied molecule that fits this criteria is DSCAM. DSCAM stands for Down syndrome cell adhesion molecule, and it has been very well studied in the insect Drosophila. It is made by fat body cells and hemocytes can be considered as the immune cells of insects. DSCAM plays important roles in phagocytosis and the subsequent elimination of pathogens, and thus it plays an important role in host defense. When we look at the gene structure of DSCAM, we can observe that there are many exons, but in the case of some exons, the exons are in the form of clusters. For example, when we look at the fourth exon, there are 12 alternative exons available. Similarly, in the sixth exon, there are 48 alternative exons available. When we look at the ninth exon, we're able to see 33 alternative exons. Finally, towards the end of the gene, we can see 2 alternative exons. Whenever alternative exons are available, then only one of those alternative exons can be used. Thus, when alternative splicing occurs following the transcription of the DSCAM gene, the mRNA that is then processed and formed can have only one of the 12 alternative exons that's found in the fourth exon, and it can have only one of the 48 alternative exons that is found in that sixth exon. Similarly, it can have only one of the 33 alternative exons that's present in the ninth exon. And finally, towards the end, it can have only one of the 2 alternative exons. This type of alternative splicing results in many different isoforms, and thus we can have different versions of the DSCAM proteins produced by Drosophila. Thus, the same protein, DSCAM, can have many variations, and the variations can be observed in the regions that are outlined in these black boxes, which is where we see the alternative exons present. With this, we come to the end of our talk, where we learned about the different characteristics of the immune system in higher organisms, followed by different mechanisms that prokaryotes and protists use to defend themselves. We then learned about the animal kingdom and the different mechanisms that invertebrate animals use to defend themselves.